Okay, guys, this is the video lecture for Chapter 12, the Genetical Theory of Natural Selection. All right, so we've talked in previous chapters about the fact that Darwin had one major hole in his theory of evolution, and that was he lacked a plausible mechanism of inheritance. We've talked about that. Darwin recognized that. It was the one thing about his theory that he never felt like he had fully addressed. And that's because no one in Darwin's day had developed a plausible theory or mechanism of inheritance, except for Gregor Mendel. Unfortunately, Gregor Mendel and Charles Darwin, as far as we never communicated regarding this really important aspect of Darwinian evolution, or the theory of evolution. So the mechanism then of inheritance was Mendelian particulate inheritance. And as I said, Gregor Mendel wrote a paper on this in 1865 and went largely unnoticed. It was published in a fairly obscure Eastern European scientific journal, and apparently no one read it, or at least they did. It didn't click. So this was sort of discovered. Mendelian inheritance was discovered in 1900 when his paper was actually read by, I believe, an English biologist. Then, in 1930, Ronald A. Fisher published a book entitled The Genetical Theory of Natural Selection, which was the first really concise attempt to sort of roll Mendelian inheritance into Darwinian selection as the mechanism through which selection operates. So Fisher's book was a pioneering work that played an important role in the ultimate synthesis or joining of Darwinian selection with Mendelian genetics. All right, so a couple of things to remember about natural selection before we move on here. First of all, selection and evolution are two different things, okay? Evolution consists of two distinct sub-processes. The first is the origin of genetic variation, and second is changes in gene pool characteristics, or allele and genotype frequencies, via selection or drift. So selection is one mechanism of evolution, but it is not evolution per se, in and of itself. All right. Second thing to remember about natural selection is that a feature of an organism cannot evolve by natural selection unless that feature has some effect on reproductive success or individual survival. So that feature has to be adaptive and heritable. Third, selection may occur between different phenotypes, but this only has evolutionary consequences if phenotypes differ in their genotypes. Remember that the environment can affect the phenotype. So then what I'm saying here is that the difference in phenotypes has to have a genetic component for those differences to be selected for or against by natural selection. All right, so let's talk about the types of natural selection that we recognize today. Well, first type of selection is called directional. Okay, so directional selection. And this is natural selection that consistently favors one genotype while consistently working against another. So the change that results, for example, could be directional in the sense that it increases body length or body size through time consistently. Guppies become more brightly colored. Beaks become longer and narrower, for example. What's going on here is that selection is favoring one extreme of the phenotypic continuum, the range of variation that a phenotype shows, and by selecting for one end of it is constantly shifting the phenotype in that direction. All right, so this can occur with quantitative or continuous variation that arises from multiple alleles, situations where you have more than two alleles, or in situations where you have multiple genes coding for a phenotypic character, what we call polygenic, because multiple genes are involved in coding for that character. Those examples are quantitative variation, and this is where directional selection really sort of comes into its own. Now, directional selection can also act on discrete variation where you only have a few phenotypic forms in a population. And discrete variation was the kind of variation that Gregor Mendel had in his pea plants. You had either purple flowers or white flowers, for example, or you had either smooth peas or wrinkled peas, for example. The reason that he had that discrete variation is because there was only one gene that coded for the character, and it came in only two forms, a dominant and a recessive. Okay. All right. Second, oops, sorry about that. Second, we have what's called stabilizing selection. Okay, so stabilizing selection is when an intermediate phenotype is selected for. That's the fittest one, or the most fit one, at the expense of phenotypes on either end of the phenotypic continuum. So this is most often associated with quantitative or continuous variation. You can think of it as, for example, heterozygotes being selected for, whereas either of the two homozygotes are being selected against. Although that's not a quantitative or continuous example, it's a discrete example, it would still work. And then third, we have what's called disruptive selection, or also called diversifying selection. Here, the extreme phenotypes have higher fitness than intermediate phenotypes, and so this is most often associated with quantitative variation as well. Here you'd have, for example, individuals on either extreme of that phenotypic continuum being selected for, and those individuals that are falling somewhere in the middle are being selected against. So maybe you have two different types of predators, one type of predator in a pond or a lake, and it feeds on individuals of a certain species that are of intermediate length. That would mean the long individuals and the short individuals would get some protection and be selected for, whereas the intermediate length individuals would be preyed upon most heavily by the predator, and we would see that phenotype decline. Disruptive selection tends to split the population up into two different phenotypic forms, and it's actually thought to be a mechanism that, if it operates long enough, could cause one species to evolve into two. All right, so let's take a look at sorry, my slides don't want to advance. Let's take a look at some examples of this from a figure from your textbook. So here we have selection effects on discrete variation. So variation that only comes in two or three phenotypic forms. This is what Gregor Mendel dealt with his pea plants, for example. All right, so in the first case here, we have what's called homozygote advantage. That's where the homozygotes have an advantage, a selective advantage over heterozygotes. So first of all, what you're looking at here, in terms of these two different shades of brown, the lighter shades of brown are individuals that are selected for. The darker shades of brown are those that are being selected against. And that's true in all these examples, okay? All right, so in this case, you can see that in the homozygotes, there are more individuals that are favored than not favored. And so in the next generation, more and more of the population would consist of the A2 homozygote that you see there. So this is sort of shifting us away from a situation where we have a relatively a good amount of genetic variability in terms of this homozygote, that heterozygote, and this homozygote, and it shifts it toward a situation where you end up with more of one type of homozygote, okay? And again, that's an example of directional selection. But it's referred to as homozygote advantage because one homozygote is being selected for at the expense of the other homozygote and also the heterozygote. In the second one here, we have what's called heterozygote advantage, all right? So, the pointer's not working. Heterozygote advantage. Here you can see that the heterozygote population is most adapted because more of them are being favored. That's the lighter color brown. Whereas a larger proportion of either homozygote in darker brown is being selected against. That means the frequency of the heterozygote should go up in the next generation, and it does. Okay. Third, we have heterozygote disadvantage. So now notice what proportion of the heterozygote population is being selected against. That should cause the frequency of the heterozygote to drop in the next generation. And presumably in this case, one of the homozygotes is being favored. So maybe this one in this case, you see its frequency went up. But you see the frequency of the other homozygote also went up a little bit as well. But the heterozygote definitely dropped in frequency or in proportion. All right, so this is an example of selection effects on discrete variation, but what about on quantitative variation? So variation that varies, that exists along a continuum between two extremes. All right, so here again we have directional, and notice the color coding. So we have this portion of the population being selected against, this portion being selected for. So if these guys are being selected against, over time you should see the average, which is denoted by the dashed line, shifting to the right. So now you can see that more of the population is shifted in the direction away from the original mean, which is what the dashed line is showing you. In other words, the curve is shifting sort of in that direction, okay, directional. All right. In the case of stabilizing selection, now you see that you have heterozygotes that are being favored, so intermediate forms being favored, and this extreme, maybe it's a homozygote, and this other extreme, maybe it's also a homozygote being selected against. So it's like chopping off the tails of the curve, basically. It tightens up and becomes more narrow, all right? That's stabilizing selection. So you have a phenotype that was already the most fit phenotype because it was already the most abundant or the most common, and now you see it's being selected for, so it becomes more common at the expense of the extreme phenotypes, which basically kind of drop out of the population. The curve tightens up. In diversifying selection, you have one phenotypic form that's being selected against, even though it's relatively abundant, and the other also being selected for. And what that can do over time, although this figure doesn't show it well, is basically cause a bimodal distribution. It can start to work against the intermediate forms here, dropping them in frequency, and over time splitting the population into two separate forms, basically two separate phenotypic forms. This is not shown all that clearly. What it could end up doing is creating a bimodal distribution like that, and so selection's working against that intermediate form, constantly driving it lower and lower in frequency. And you could end up with, population 1 here and population 2 that, if given enough time, could evolve into separate species. All right, that's why this is called diversifying selection, because it could produce more species from an original ancestral species. Okay. All right, so now let's begin to talk about fitness. So fitness is the mean lifetime contribution an individual with a particular genotype makes to the population in the next generation. It's usually described in terms of reproductive success or the mean number of offspring or eggs descended from one individual that survive. All right, so let's look at an example here. Let's say we have two genotypes. We'll just call them genotypes A and B in a population, okay? And let's say that A, genotype A, has an egg survival rate of 0.05 or 5%, all right? That means that 5% of the eggs survive to reproductive age. Okay, and let's say that each individual that has genotype A produces 60 eggs on average over their lifetime. All right, then the fitness of A equals 0.05 times 60, which would be 3 offspring. So it's going to produce, on average, 3 offspring to the next generation. All right, so the number of offspring then that you see right there that it contributes to the next generation will be the per capita growth rate. So that's R, per capita, growth rate. All right, per capita growth rate. That's R. Okay. Now, let's say that genotype B, on the other hand, draw a line here, genotype B has an egg survival rate of 0.10, all right, or 10%, and produces 40 eggs in a given generation, okay? Well, its fitness, then, as we did before, equals 0.10 times 40, which equals 4. Okay, so R in this case equals 4. Okay, so which of these two, genotype A or B, has the higher fitness? B, because it has a rate of growth of 4. All right. Okay, so let's take a look at this. As we said, genotype B has egg survival rate of 0.10, and each individual produces 40 eggs, so its fitness is 4, right? Rate of growth is 4. So if we sort of extrapolate that out over time, over a series of, let's say, 5 generations, you could see that genotype A, producing 3 individuals per generation, is just not competing in terms of number of offspring produced with genotype B, which can produce 4. So the more fit phenotype is the one that increases in numbers fastest. And certainly genotype B is more fit because individuals that possess it produce more offspring each generation, and thus over time. Okay, so our per capita growth rate, again R, represents what we call the absolute fitness of a genotype. Okay, the absolute fitness is telling you in absolute terms how many offspring per generation that genotype should contribute to the next generation. Okay, we can contrast that with what's called relative fitness. Relative fitness is W, okay, or W sub I, okay, relative fitness. So relative fitness of a genotype is its fitness in comparison to a reference genotype. So the reference genotype would have a fitness or relative fitness of 1.0, and the relative fitness of other genotypes then would be somewhere between 0 and 0.99. Okay, so we're assuming here that, let's say, genotype A is the most fit. That one has a relative fitness of 1.0, and any other genotype in that population would therefore have to be less fit and would have a fitness of between 0 and 0.99. All right, so think of the relative fitness of the most fit genotype as 100%, and the others must be between 0 and 99% as relative fitness, okay? All right, so we talked about genotype A in our previous example having a fitness of 3, an absolute fitness of 3, compared to B, which had an absolute fitness of 4. So in terms of relative fitness, W sub A, which is the relative fitness of genotype A, would equal 3 over 4, or 0.75. It's only 75%, or 3/4 as fit as genotype B. Genotype B, right, would have a relative fitness of 4 over 4, or 1.0. Okay. All right, so this then brings us to a really helpful topic, or concept rather, called the coefficient of selection. Let me erase this real quick. Okay. Coefficient of selection. All right, so the coefficient of selection is S, and it is the difference between the fitness of a particular genotype and that of a reference genotype. So in our example with genotypes A and B, where they differed by 0.25, right? So remember that A had a fitness of 0.75, and B had a fitness of 1.0. The coefficient of selection is the difference between those, the fitness of those genotypes. So S then equals 0.25. All right. Okay. So this tells us how intense the selection pressure is against the less fit genotype, or conversely, the selective advantage of the more fit genotype. All right. So now we're going to get into talking about genetic models of selection. Okay, so models of selection. And these models generally, or in particular, focus on what's going on at the genetic level. Okay, so we've talked about directional, we've talked about disruptive, we've talked about, disruptive, and we've talked about stabilizing. Okay, so now we're going to look at those from a genetic standpoint, the standpoint of the gene pools and the frequency of genotypes within them to see what's really going on. Okay, so in a single locus two allele system where the heterozygote has a fitness that's precisely in between the two homozygotes. So in other words, if we had a situation where you had A1 being more fit than the A1, A2 heterozygote, which is then in turn more fit than the A2 homozygote, and the A1, A2 heterozygote is exactly in between, so intermediate between the two homozygotes. Question is, can we determine at what rate an advantageous allele increases in frequency? Excuse me. Yes, we can, with a little bit of math. All right, I know you're excited. Okay, so the frequency of allele A1 is denoted as P, and the frequency of allele A2 is denoted as Q. And we've already seen that back when we talked about Hardy-Weinberg equilibrium. P squared plus 2PQ plus Q squared equals 1, right? Okay, so we have an equation then that calculates the rate of directional selection for an advantageous allele. In other words, it's going to tell us or help us to figure out how quickly an advantageous allele that's being selected for can increase in frequency over subsequent generations, right? So let's say we're talking about allele A1, so P in other words. All right, our formula then is the change in P, so Greek letter delta means change, think about chemistry, right? Change in P is 1/2 our selection coefficient times the frequency of P times the frequency of Q, divided by 1 minus the selection coefficient times Q. All right, so that equation then calculates the rate of directional selection for our advantageous allele, P. All right, so again, just so we have all this down, P is the frequency of the A1 allele, Q is the frequency of the A2 allele, and S equals our selection coefficient. All right, so the difference in the force of selection between the advantageous allele and the less advantageous allele, between A1 and A2. Okay, so let me erase this here real quick. Sorry, it's a little bit touchy. All right, and let's start again. So let's assume that allele A1 is initially present at a frequency of 0.08. In other words, 8% of our gene copies are allele A1. All right, that means that A2 must be, if we only have two alleles, A2 must be frequent or present at a frequency of 0.92. 92% of our alleles must be A2. All right, let's say that genotype A1, so a homozygote for A1, has a relative fitness, so W, of 1.0, and genotype A2, relative fitness equals 0.89. All right, at what rate per generation does A1 increase in the population? So the first thing we have to figure out, so step number 1 is our selection coefficient, okay? Well, the selection coefficient equals the relative fitness of A1 minus the relative fitness of A2. So that equals what? 1.0 minus 0.89 equals 0.11. All right, so that's our selection coefficient. All right, second step is just to plug all these numbers then into our formula that we saw in the previous slide. So again, to remind you, the frequency or the change in the frequency of A1 allele, in this case P, equals 1/2 S PQ over 1 minus SQ. Okay, so that's going to equal 1/2 0.11. That's our selection coefficient, times the frequency of P, which is 0.08, times the frequency of Q, which is 0.92. That's a 9, sorry. And all of that divided by 1 minus 0.11 times 0.92. All right, so you work out that math, you get 0.004 over 0.8988, and that equals 0.0045 with a little rounding. So the frequency then of A1, which initially was 0.08, A1 frequency in generation 2 equals 0.08 plus 0.0045, and equals 0.0845. So our frequency of our advantageous allele has gone up to 0.0845. Now, that also means that the frequency of A2 allele, which is being selected against, is going to decrease by the same amount. It goes down by 0.0045, so it ends up being 0.9155 at the end, or in the next generation. So allele A1 has gone up in frequency, A2 has gone down in frequency by how much? That much. All right. So that's why it's useful to understand the selection coefficient, okay? You got to remember that our change in P equals the negative change in Q. The magnitude of the change is the same, the direction of change is the opposite, up in one, down in the other. Okay, so an important result of this model is that the change in an advantageous allele is positive as long as the selection coefficient is greater than 0. Even a selection coefficient of, say, 0.000001 will eventually result in fixation of an advantageous allele as long as no other evolutionary forces like drift or non-random mating or migration intervene. That's a really important implication of this model here. As long as selection coefficient is positive, it doesn't matter how small it is, eventually it will drive an advantageous allele to fixation as long as something like drift, let's say, doesn't knock that allele out of the population. All right, and that's how you end up with really exquisite adaptations like this. So this is a cryptid katydid, and what you're seeing here basically is an animal that is marvelously camouflaged to look like a leaf, right? Look like a dead leaf on a branch. So you can see, for example, here's the body, here's the abdomen of the animal. You can see the hind leg there. There's the eye, that's the beak, right? This is the head, that's the thorax here. And you can see that large extension off the back of the thorax. It looks dead like a dead leaf. So the fact that even minute advantages will cause alleles to become fixed by selection explains the seemingly perfect adaptations of many organisms. In this example, it's easy to suppose that a less well camouflaged individual might be about as well protected from predators. However, as long as any new mutation, such as another blemish on the exoskeleton, for example, provides even the most minuscule advantage, a slightly higher selection coefficient, it will be selected for and may eventually become fixed. All right. Now, just how long it takes for an advantageous allele like that to become fixed depends on a number of things. First of all, it depends on the initial frequencies of P and Q, so of our two alleles, okay? It depends on the selection coefficient, and it depends also on the degree of dominance, okay, degree of dominance. So as an example, a completely dominant allele will increase the fastest because it is expressed in two genotypes. A completely dominant allele, say A1 for example, is expressed as an A1 homozygote, but also as an A1 heterozygote. If allele A1 is completely dominant, A2 has no effect on the phenotype of this heterozygote. So the phenotype of the heterozygote looks just like the A1 homozygote's phenotype. And so selection cannot see that recessive is there. All right, so this selection sees this one just like it sees that one, and it would favor them both. They both have the same selection coefficient. Now, an incompletely dominant allele will increase less quickly because A1, A2 in this case would have a phenotype somewhat different than the A1 homozygote. So that A2 then would have some minimum effect at least on the phenotype of this individual and would thus reduce its fitness. Okay, so these A1 alleles then that are in heterozygotes would not be passed forward into the next generation at the rates at which the A1s in the homozygotes would, and so they would not contribute as much in the next generation to the increasing frequency of A1. So in this case, where you have incomplete dominance, the A1 does not increase in frequency as quickly because the heterozygotes that the A1s can also be in, like you see here, would have lower fitness. All right, so let's take a look at that. So kind of zoom in on this top figure here first. All right, so notice first of all that we have our initial frequencies of 0.01, right? So this is the frequency of the advantageous allele. Initially it's at 0.01, so almost zero. It's way down here, okay? And then we have time going forward up to 100 generations. All right, so even if a recessive allele is advantageous, it takes a very long time, as it says here, to increase in frequency if it's very rare initially because very few homozygotes are formed. So if you have a population that consists of 99% A2 alleles and 1% A1, how likely are you to get A1 homozygotes? Not very. They're going to be very rare, and so selection would act on them very slowly because they're just not that common. So it would take a long time for that A1 allele to increase in frequency if it's being selected for. All right, but what if A1 is dominant, completely dominant, okay? Then you've got it being selected for in the A1 homozygote, but also in the A1 heterozygote. So it could increase relatively quickly, okay, and eventually maybe go to fixation. What if we have a situation of intermediate dominance where A1 is not completely dominant? So again, as I said in the previous slide, you have A2s, heterozygotes, that are probably being selected against, and so that's taking A1 alleles out of the population. Well, in this case, selection is only favoring the A1 homozygotes, so it takes longer for the allele to ultimately increase and go to fixation. Okay. All right, now let's look down here at the second figure. Now notice that we have our allele present at a much higher frequency initially, say 10%, 0.10, okay? Now you see that we have the same basic pattern in the case of complete dominance and intermediate dominance as we did up at the top, okay? But now you notice that in the case of A1 being recessive, all right, it actually can take off quite a bit faster because it's present at a much higher frequency here, 10%, than it was up here, okay? So the initial frequency of an allele is an important consideration for understanding ultimately how fast it could go to fixation if selection is favoring it. All right, an allele that's a bad thing to have, what we call deleterious. Okay, let me write that word down because it's kind of a strange word. A deleterious allele, one that's being selected against, can be eliminated a deleterious recessive allele will be eliminated very slowly once it reaches low frequency because it's far more likely at that point to end up in a heterozygote than in a homozygous recessive genotype. So let's say that A2 is a deleterious allele, bad thing to have. You'd have selection certainly, operating against A2 homozygotes. But A1, A2 heterozygotes, especially in a case of complete dominance, selection sees that genotype the same as it sees the A1 genotype that's being selected for. Oops, sorry, go back. So this one's being selected for, but in the case of complete dominance where selection does not see that A2 because it has no effect on the phenotype, this genotype is also being selected for at the same rate as our A1 homozygote. So the point here is that once selection has whittled A2 down to such a low frequency, it's far more likely to end up in a heterozygote than it is as an A2 homozygote because you need two copies that have to come together. one sperm, one egg, both having the same allele to make an A2A2 homozygote. That's not as likely as a heterozygote. Even if A2 is rare, you've got A1 being very dominant, or very common rather, so mostly you'll get A2 pairing up with A1, and selection sees this genotype in the same way it sees that one. So these A2s here can kind of hide out. That's the whole idea behind a carrier, right? A carrier carries a recessive allele that selection can't see, and the only way that that allele can be acted on by selection is if it happens to come together in the next generation with another copy just like itself, giving you this, that selection could then operate against. But this is a mechanism for maintaining genetic diversity in a population where completely recessive alleles can't be seen by selection when they're at low frequency because they're bound up in heterozygotes. All right. So I think that's been about a half hour or so. I'm going to go ahead and stop here. The next concept is about adaptive landscapes, and we'll use that to kick off part 2 of this Chapter 12 lecture video.